sympathetic. Take a break. It'll only take about 40 minutes, 45 minutes to finish up sympathetic. Take a break. Let you guys review and study for the quiz. And then spend about 35, I think that's what it took me this morning, 35 or 40 minutes getting the eyeball structure underway so that on Thursday we can talk about the mechanisms of vision and how we bend light to make images and stuff. And it will be easier to follow that without trying to do all of that on one day. And then we'll look at the models of the eyeball. And then Friday we have the eyeball dissection along with the ear structures. Okay? Yes?
The quiz, the online quiz,
No, because it was extra credit, so I didn't change it. And I looked at all the questions and they were all on Tuesday's material last time. Okay, so looking at contrasting parasympathetic and sympathetic. Up here is a list of some of the major effects of sympathetic activation. And the first two really don't have anything to do with what we typically look at for the autonomic nervous system. Anybody remember what RAS stands for? Or it can be described as RF, reticular. Yeah, the reticular formation or the reticular activating system. So this is a neurotransmitter that's involved in your conscious awareness, waking up, feelings of energy and euphoria, and sensitivity to pain stimuli, which is involved in the pain processing in the brain. And then 3, 4, and 5 are going to be components of our typical autonomic response that we think of in the off the spinal cord and the thoracic and abdominal cavities. Anybody remember the group of cells called in the brainstem that release a lot of norepinephrine under stress? It was in the slides when we went over the neurotransmitters and their pathways. And remember, what amino acid is norepinephrine derived from? Tyrosine. Okay? So when tyrosine is converted into dopamine, remember it forms a black pigment, and we call that the substantia nigra. When it's converted into norepinephrine, it forms a dark blue pigment. So remember what the Latin term for the blue spot was? Ceruleus. Close, yeah. Ceruleus, the locus. Ceruleus is a group of nuclei in the brainstem, pons, that releases norepinephrine under stress. And I'd mentioned that in our pathway. So in addition to our sympathetic, our thoracolumbar, lateral gray horn region, I wanted you to recognize locus ceruleus as a brainstem group of cell bodies that release norepinephrine. Before we move on and compare some of the functions of the sympathetic versus the parasympathetic, as a neurotransmitter, we talk primarily about norepinephrine. But it's often mentioned norepinephrine / epinephrine. Another term for epinephrine, more of a layman's term, is adrenaline. And that has to do with its source. So forming a little Napoleon's cap sitting on top of the kidneys are a pair of glands known as the adrenal glands or suprarenal. And the outer portion of it is going to release steroid hormones, stuff like cortisol, if you've heard of cortisol. But the cells in the middle are embryologically the same as postganglionic sympathetic neurons, except they don't have long axons. And so those are the cells in this illustration right here. Find it really fast. That are illustrated here and sending their chemical into the blood. The adrenal gland is an endocrine organ. So these cells in the adrenal gland are derived from the same cells that would have formed neurons, only they don't have long axons. And so they send their chemical into the blood, so instead of being a neurotransmitter, it's a hormone. And that's the primary source of epinephrine, okay? Which is very similar, but not quite identical to norepinephrine. But they bind to the same receptors often. Okay? So when I say epinephrine, norepinephrine, this is the most common source of epinephrine, okay, or adrenaline. All right, now let's look at parasympathetic. Effects and sympathetic, just to kind of get an overview. And remember I used the term dual innervation. So many organs will have postganglionic neurons releasing either acetylcholine or norepinephrine. And for the most part, these effects are opposite. All right? So let's kind of start at the head and move down. So parasympathetic effect on the pupil is going to be, this is just a summary before we move back into sympathetic, is going to be constriction of the pupil, whereas sympathetic is going to be pupillary dilation. And remember, sympathetic fibers will get to the pupil by coming off the superior cervical ganglion and following the internal carotid artery up into the skull, and then following blood vessels with the optic nerve out to the eyeball. Okay, so that's, again, following blood supply is how it gets out there. Close-up vision. Due to rounding of the lens. And this is distance vision due to flattening of the lens. All right, secretion of the glands in the face. So we're going to have increased tears and increased saliva. Again, opposite effect. So decrease glandular secretion, fewer tears, less saliva. And again, the process here is thought to be vasoconstriction, so there's less fluid going to these glands, so they can secrete less. As we move into the thoracic cavity, the heart rate without any nervous control at all is about 100 times a minute. That's not our normal heart rate. So remember, parasympathetic maintains homeostasis. It's rest and digest. So the effect of acetylcholine on the heart rate is going to be to decrease heart rate. Sympathetic is going to be increased heart rate and increased ventricular contraction. Parasympathetic really doesn't have an effect on ventricular contraction, but you don't need to know it to that level. For the respiratory system, the airways are constricted. By the parasympathetic. For sympathetic, airways are dilated. And then for the digestive tract, we call this state when you first, a few hours, 1 to 2 hours after you eat, is called the absorptive state. And so this is increase in digestive activity. So we have an increase in peristalsis. We have an increase in glandular secretion, so enzymes. Digestive enzymes and insulin. So we have too much of our cholinergic drugs, we're more likely to have diarrhea because we're going to increase that peristalsis more than normal. Too much of our sympathetic drugs, we're going to have a decrease, so more likely to have constipation. So this is the post-absorptive state. This would be anywhere from 2 to 6 hours or longer. So a decrease in peristalsis, a decrease in glandular secretion. So a decrease in digestive enzymes, and now instead of insulin being up, we're going to have an increase in glucagon. Because glucagon releases sugars. So the parasympathetic effect is to lower blood sugar right after you eat, especially if it was a donut. And parasympathetic effect is to increase. That's glucagon over here. I'm kind of writing quick. Okay, urinary bladder. There's two things to the bladder. When you go to the bathroom, you need to contract the bladder, but for urine to leave the bladder, the sphincter has to be released. So it's going to be relaxation of the sphincter, contraction of the bladder wall. This is going to be the opposite. Contraction of the sphincter. Relaxation of bladder wall. All right, so in this list of activities, all the organs are innervated by both parasympathetic and sympathetic, and the effects are the opposite. All right. What would be the name of the receptor, a nicotinic or muscarinic receptor on each of these muscles or glands for parasympathetic? Do you remember? The nicotinic receptor, well, what's a neurotransmitter? We'll do something that you know. Acetylcholine. The neurotransmitter is acetylcholine from the postganglionic neuron, because we know that for both it's going to be acetylcholine for pre, all right? So the receptor along here is described as muscarinic. The nicotinic is found on the postganglionic neuron itself, released between the pre and the post. So these are muscarinic receptors. Are they ionotropic or metabotropic? These are all metabotropic. The ionotropic is in the ganglion. So the ganglion is nicotinic and ionotropic. These are all metabotropic. Right? That's how we can have either excitatory or inhibitory effects. So we have an inhibitory effect on the heart rate. Almost everything else is excitatory. We have an inhibitory effect on the sphincter of the bladder. All right, and G proteins are what allow us to do that by activating or inactivating the cyclic AMP or the inositol triphosphate component. So these would be nicotinic. And remember, it's true for both sympathetic and parasympathetic. Acetylcholine is released, and they're nicotinic receptors, whereas at the organ, for parasympathetic, it's acetylcholine. Okay. All right, sympathetic is going to be norepinephrine. Primarily. Still going to be acetylcholine and nicotinic at its preganglionic. But at the organ, it's going to be norepinephrine. And as we'll see today, we talk about two different types of receptors, alpha and beta. And that's what we're moving towards today. Now, these are opposite effects. Okay? We have one system where sympathetic and parasympathetic actually work together. Anybody remember which one that is? Reproductive. Okay? So can I erase what's on the board here and just do the reproductive system components?
So the alpha and the beta, those are receptors?
Receptors, types of receptors for norepinephrine. This piece of paper under one of the legs.